introduction of NIPGER. So as you know, NIPGER is National Institute of Plant Genome Research, right? We are supported by Department of Biotechnology, okay? And the major area that we work and the major tools and techniques for research purpose we use is molecular biology and plant biotechnology, alright? So welcome again. And so our institute was announced in 1997, alright? And then we became functional institute in 1998. And initially this setup was established in the premise of Jawaharlal Nehru University, okay? So this was known, so like you see, these 16 institutions, right? So these are all supported by Department of Biotechnology, and NIPGER is one of them. Okay? सभी institutions के जो mandates हैं they are different. For example, our mandate is to do research in the frontier areas of plant science. Likewise, there are other institutions like the National Institute of Immunology, ICCB, TIST, right? Across the country, they have different mandates and they are working according to their mandates. So if we see the location of NIPGER, it's a wonderful locality in which we are surrounded by premier institutions like we have JNU, just we are sharing the wall and the campus of JNU itself. Then IIT Delhi is nearby. We are sharing boundary of ICCB, okay, National Institute of Immunology is there, and there are others, right? So why I'm telling you about the location and locality? Because it is a wonderful place to be here because you have wonderful chance to attend and build a strong network in other institutions. You can attend here different programs, workshops, trainings, and your students also come to attend our program. So it is a wonderful opportunity to build your network and also get to know what kind of research and different kind of opportunities that is available here. This completion and difference. Your degree is awarded by Jawaharlal Nehru University. So our PhD program is basically affiliated with JNU. Okay, so what are the major research areas that we are working in? Climatic factors, है ना? Temperature हो गया, drought हो गया, salinity हो गया, right? तो ये सब क्या हो गया? Abiotic stresses, है ना? तो इस तरह की जो factors हैं, चाहे biotic है या abiotic है, तो different labs as per their requirement of the projects and objectives, they are working. Then we also work on plant microbe and plant insect interactions. So the plants हैं, they also try to defend themselves, है ना? चाहे वो pathogens है, चाहे insects है, चाहे यार biotic है या abiotic जो factors है, plant जैसे हम लोग भी, we also try to protect ourselves, right? चाहे बाहर की धूप है, गर्मी है, dust है, या फिर pathogens, like अभी कोविड सिचुएशन था, तो what we were doing, we were protecting by wearing mask, है ना? तो ये हमारा एक mechanism है to protect ourselves. But plants are not movable, so they have very different kind of mechanism to protect themselves, है ना? तो what we try to understand कि उनके अंदर ऐसे क्या mechanisms हैं, क्या signaling molecules हैं, क्या developmental processes हैं, जो अलग-अलग situation में वो survive करते हैं, है ना? So there are different groups like Dr. Jyoti Lakshmi is working on that. Besides that, we have computational biology, okay, and nutritional traits in crops that self-explanatory. कि जो भी crops हैं या pulses हैं, उनकी nutritional values में improvement करने की कोशिश करते हैं. For example, हम लोग का एक chickpea का improved variety है, which has almost 10 to 11% higher protein. ठीक है? So and besides this, we also have one new group, Dr. He will join us for a scientific lecture today. So he has started working in the structural biology. Okay. So coming to the publications, our faculty and scientists from the institute, they are publishing their scientific findings in various reputed journals. These are just the representative photographs of the various journals where our research findings has been published. All right. So our faculties, I'm not going individually with the name of the faculties, but our faculties has been awarded with the various reputed that include Padma Bhushan by the government of India. And this was given to our founder, Dr. Professor Ashish Chitla. Then S. Ramachandran National Bioscience Award by DBT. Nasi Reliance Industries Platinum Jubilee Award, National Women's Bioscientist Award, and IYPE, Innovative Young Biotechnologist Award by DBT. And our faculty, most of them in fact, they are members of various academies, right? So what are academies? You guys know? साइंस अकेडमीज होते हैं, right? तो क्या है, कंट्री में बहुत सारे इंस्टीट्यूशंस हैं, रिसर्च सेंटर्स हैं, है ना? इसमें हमारे जो भी फैकल्टी या साइंटिस्ट ये जो काम करते हैं, तो इनको अगर इनके एक्स्ट्रा ऑर्डिनरी कंट्रीब्यूशंस होता है, अचीवमेंट्स होता है, एक सोसाइटी टाइप की अलग अलग टाइप की बनाई गई, तो उसको वो क्या है, अकेडमी सेंटर, ठीक है? जैसे इंडियन एकेडमी ऑफ साइंसेस। तो इसमें क्या करते हैं, दे गिव मेंबरशिप, इफ यू रियली डू वेल they honor you with their membership or as a fellow, and where then you and your work is being promoted across the country and ठीक है? तो इस तरह के बहुत सारे academies हैं, for example National Academy of Sciences, that is NASI, Indian National Science Academy INSA, Indian National Young Academy of Sciences INIAS, ठीक है? and इसके अलावा और भी बहुत सारे important जो fellowships हैं वो भी हमारे faculties को award हुआ है, for example National Fellowship by DST SCRB, Government of India, Swan Agency Fellowship by DST, Government of India. Alright? So, now what kind of facilities we have at NIPGER? Like you see, so I have just written few names, and so we have various facilities. So, first one is central instrumentation facility. So let me just brief you. So in central instrumentation facility, actually all the major instruments and basic instruments are here, and which are available for the usage of all the researchers, all the labs, anytime. In fact, in our central instrumentation facilities and other facilities, if any of you from other institution would like to conduct or perform some experiments, so you can indent us, you can send a request, and if required, on the charges basis, your experiment can be done, right? So I'll come to the other facilities. So likewise, all these are different facilities that NIPGER has. So I'll just briefly walk through the confocal microscopy facility. Okay. So so how many of you know a little bit about the confocal microscopy? Microscopy के बारे में? Just in brief. अच्छा, माइक्रोस्कोपी क्यों करते हैं? Why we माइक्रोस्कोपी क्यों करते हैं? Why we use माइक्रोस्कोप? तो सिंपल सी चीज है, अगर कोई भी बहुत स्मॉल ऑब्जेक्ट है ना, and if you want to study that, तो अब अगर स्मॉल ऑब्जेक्ट है, you cannot see with your eyes, right, physically. So what we do, we use instrument known as microscope, है ना? तो वो क्या करते हैं, उसको मैग्नीफाई कर देते हैं, and when these images are magnified, तो आप एक proper structure study कर सकते हो, right? क्योंकि आपका उसकी visibility magnify हो जाती है, right? So we have confocal microscope. This is one of the type of microscope. ठीक है? इसमें आप क्या करते हैं, different layer. आपने CT scan images देखे हैं, है ना? ब्रेन का देखा होगा, vertical sections में भी capture करते हैं, horizontal भी capture करते हैं, है ना? और किसी भी layer में if you find any kind of deformities, तो उन लेयर्स के बेस पे यू आइडेंटिफाई कि ब्रेन में कुछ चोट है, स्वेलिंग है, काइंड ऑफ थिंग, राइट? सिमिलरली आप confocal microscopy में क्या होता है, we use lasers, ठीक है, like so you see here in this case neon and neon lasers that are used to capture images of various fluorescent dyes. For example, dyes होते हैं जैसे GFP, green fluorescent protein, YP, yellow fluorescent protein. So with the help of these dyes, different images capture किए जाते हैं, and that helps you to study the structure and function. So for example, if you see this Nicotiana benthamiana plant, ठीक है, हम लोग ज़्यादा डिटेल में नहीं जाएंगे, and so अगर हम इसको इन्फिल्ट्रेट करते हैं एग्रोबैक्टीरियम के साथ, है ना, उसके अंदर तो you see these are fluorescent GFP, यानी green fluorescent proteins, ठीक है? So I'll just walk you quickly with the other facilities. Then we have next DNA sequencing facility. DNA sequencing facility में what you do, you prepare your sample, वेरीफाइड डीएनए जो सैंपल होता है वो हम लोग हैंड ओवर करते हैं, दे प्रोसेस इट एंड देन गिव यू डेटा। ठीक है, सो डीएनए सीक्वेंसिंग सुना है आप लोगों ने, है ना? क्या करते हैं हम लोग डीएनए सीक्वेंसिंग? तो एज द नेम इंडिकेट, डीएनए का सीक्वेंस स्टडी करते हैं ना। अभी कोविड का पैंडेमिक टाइम था, दैन यू रियलाइज कि यहाँ पे कोविड का एक नया वेरिएंट आ गया, फिर नेक्स्ट आ गया, राइट? तो कैसे पता चलता है? तो वो क्या हो रहा था, उसका सीक्वेंसिंग करते थे। ठीक है, बट ये जो कोविड वायरस था, इसके अंदर जेनेटिक मैटेरियल एट डिफरेंट पोजीशन, है ना, एंड दैट टेल्स यू कि ओके इसके अंदर कुछ म्यूटेशन